Hi there. This is the lecture series, an overview of Focused Ion Beam Microscopy. I am Dr. Ben Britton. This series was created with the help of a range of people at the University of British Columbia, Imperial College London, Rouen, and Tescan. In this lecture, we're going to focus on 3D FIB tomography approaches and analytical 3D FIB. We'll see this for a few different examples in here. I hope you've had a chance to see at least a few of the other modules because they really give you quite a good lead-in and specific challenges that we had to overcome to make this work. So, within this series, when we're doing analytical 3D tomography, broadly speaking, this can be viewed as slice and view approaches. So when we do a slice and view approach, we're going to use this akin to reading a book, where page by page or slice by slice, we use the FIB to remove a layer and produce a new fresh surface. For every single fresh surface that's produced, we're going to then use the scanning electron microscope to analyze point by point. And we're going to have a series of pixels in each slice that will then become voxels as we start to reconstruct the three-dimensional volume that's presented. Now, importantly in this approach, we can effectively do this characterization using a variety of analytical methods. Of course, it depends on the configuration of your instrument and the sample and the type of information that you want to obtain, and of course the cost of the data set you'd like to access. So in this case, we might want to characterize each slice by looking at perhaps secondary electron imaging, either using the ETD detector or some in-lens combination. We could use backscatter electron imaging, or we could do chemical spectroscopy with EDS or EDX. We could do crystallographic analysis with EBSD. And given that we just previously in the last lecture talked about site-specific methods, we can also enable this free surface to be analyzed with great precision with wide angles, and that can be done using a lift-out approach, as we'll show a bit later, which is super important in particular for EBSD methods. So let's just have a look at the simple part of a xenon FIB tomographic experiment. So I'm just going to orientate what we've got. This is effectively going to be a short video where we're going to have the SEM pointing at the sample. The sample is sitting in here. This is the chamber scope image. We have the FIB that's pointing over here. This instrument is equipped with effectively a double tilt stage. We call it the rocking stage. This enables us, the first tilt enables us to tilt between the FIB and the SEM columns. The second tilt enables us to rock the sample with regards to the FIB to address curtaining artifacts. You can see one of those curtain artifacts towards the bottom of this area. It's that black line, whereas there's this shadow associated with some, say, precipitate or interface that is causing some slight issues in our data set. Now, on this example, we have effectively a surface cap layer. We have a steel substrate. This blue box is effectively 200 micrometers by 200 micrometers. And this effectively is a backscatter electron image that was collected off the free surface. Now I'm going to let this short video play. It's sped up very slightly. But what we'll see effectively is that we now can do a series of effectively FIB mills to remove that layer. And we're going to use this at, say, 3 different angles to try to reduce the impact of those curtains. And so that enables us to remove the material without having too much of those curtaining artifacts. Once we've done a FIB slice, we can then align it towards the SEM, and we can scan to reduce effectively a 2-dimensional picture of that free surface. And then we repeat this again, and we're going to do the FIB effectively multiple times. And if we highly automate this process, we can be very lucky, and we can effectively do a tomograph where we now have a 200 micrometer by 200 micrometer field of view for each slice, and each of those slices now has effectively this number of points. We can control effectively the pixel size within the slice by choosing the resolution and field of view size, and we can choose the thickness of how much material we have removed. If we do this multiple times, we can remove here 668 slices, and the total data collection time for this backscatter electron micrograph was about 42 hours of FIB time for here. Here we've optimized it to do relatively quick imaging, so 1 minute and 10 seconds per slice. We've done the cutting at about 2.5 minutes per slice, as you saw. But this is super powerful. We've seen this video actually before in the series, where we now have that slice and view that enables us to basically digitally remove each layer and look at the evolution of that microstructure in 3 dimensions. This is pretty well done to reduce curtaining, and this was using the rocking plus the xenon fib to enable this microstructure to be digitally reconstructed. What's super nice is that in this setup it's very well aligned, that you can't really tell which direction each of the slices were reduced. There's not too much of a misalignment error in this data set. It's quite stable for how this is operated. And that's particularly how our microscope has been configured to enable this to be easy to do and enable us to reduce the amount of digital alignment steps we have to do in our data sets. So, one of the important aspects is once you've got the raw data and it's well aligned, the slices are put together, that tomograph looked very pretty, but actually the trouble is what do you want to do with that data? And so here, effectively, 2-dimensional data is quite nice. You can print it out and point at different features that are interesting. 3-dimensional data becomes super annoying because you might want to look at the connectivity or porosity. So what you traditionally have to do is to take the raw data and then you have to perform segmentation. Now, fortunately, there are a number of different software packages, for instance, Aviso and Dragonfly, that have significant steps forward to make this relatively routine and easy to perform. Previously, what you might have to do is to color in every single slice. We'll see how that was done in an example data set from Dr. Angela Good that was kindly given to me. Once you've done the segmentation, you can then do a volume reconstruction, and then the question is, what do you do with this volume reconstruction? You might put it in a model, like a computational fluid dynamics approach, or you might start measuring features and regions within that data set. So I just want to show a really nice example from Dr. Angela Good. This is part of her PhD work at Imperial College in London. And so Angela worked on effectively looking at biological systems and specifically looking at cobalt chromium wear debris associated with prostheses. So this is a tribo problem that creates some nanoparticles, and then the question is where do they sit in the microstructure of the biological system. So Angela did some beautiful fib-SEM tomography using a gallium fib in this example, and so here we can see that slice and view through the gallium fib. This was reconstructed, I think, using Aviso with this graphic. So we can see those slices, and then Angela did some very nice segmentation to pull out a particular phase that then enables us to get this 3-dimensional tomograph that then can be rotated to reveal the structure that's presented in this micrograph. And so that starts to be quite powerful once the data has been properly segmented. And we can effectively look at it across different angles, and we can of course slice through and cut, make several virtual cuts to look at the structure. We can label alternate phases and how they intersect with that microstructure, and so that again becomes very powerful to look at 3-dimensional objects, for instance their spacing, aspect ratio, size, shape, interfaces, connectivity, etc. in this. We can see another example from Dr. Good's work looking at the 3D orientation of collagen fibrils in a lymph node conduit. And so in this system we effectively have lymph node conduits and they contain collagen fibrils that are in this sort of bundle, the so-called conduit. This is a transmission electron micrograph, so this we can see a 200 nanometer or 0.2 micron scale bar, and we can see those features. So Angela took these sort of ideas and basically collected a series of BSE images. This is a contrast inverted of this from the stack, and then performed fib-STEM, and you can see now the sectioning through that object. That's super nice. You can start to see how the structure and in your brain, depending on how good 3D imaging is, you can start to see and imagine how that is connected through the object. Now, some small alignment issues, but it's pretty good data, especially given that it's a biological system. And this was collected probably about 12 years ago, 15 years ago now, from when I was kindly shared this. So super nice. It's been registered and aligned to give you this structure. Now, what's super nice is that once you've got that data, you can find a common region of interest that contains the collagen fibril cluster, and you can segment using, I think this is done in image J or similar, to effectively identify those features. So you have a small region of interest. But what's really nice is that now you can go through and reconstruct how those fibers are connected slice to slice. So this effectively, again, is that aviso slicing of the labeled data. And then we'll see the object rotate, and we can now draw out that three-dimensional bundle cluster. And we can see, for instance, how continuous some of those fibers are and how tortuous some of the other fibers are. And this is really starting to give you an idea where that three-dimensional data is particularly useful for looking at, say, fibrous structures that can be quite hard to interpret from just a single 2D slice. Certainly if you cut this orthogonal, it would be quite challenging for you to see that structure. We can also look at this, so that was biological systems. I can also show some work from Dr. Ruth Birch. This is looking at steel samples. And here we're now going to expand the approach to include some analytics. And so in this case, we're going to use electron backscatter diffraction. So we're going to use the Amber X system. We're going to do some rocking polish. We're going to do EBSD on each slice. And then we're going to effectively reconstruct the data for this structure. So here we see how the microscope is operating. So we're going to do the fib process, and the sample moves around. It's been set up on a long post, and the rocking has been aligned. So we're fibbing away to remove each surface, get a nice finish. We then produce, insert the EBSD camera. This has been sped up, of course, just to protect the camera between fib slices. We collect an EBSD map, where we show the crystallographic structure. We're then going to remove the camera. We're then going to fib a layer, and we're going to repeat this across multiple slices, and now you can start to see the building of that tomogram. You can now start to see how that crystallographic microstructure is being swept through. And this is actually quite a large field of view as presented in this tomographic data set. So this is for the EBSD. This was done on the edge of a sample. We've actually cleared some of the material out of the way so that when the tomograph, this slice is removed, the redeposition is not getting and causing us issues by getting stuck back on the sample here. There's also a protective cap layer on top as well. So we've seen this, we've seen the steel sample as presented, but we can also look at EBSD data to start to see the slice and wipe of the EBSD data, where we can start to see that microstructure in 3 dimensions. Here we've just got the IPF with respect to the z-axis. Different colors give you different grain orientations. But we can also do effectively 3D EDS, and so this is an example of some tin bismuth. And this is just a beautiful example to show that structure. We've seen this video previously in an earlier lecture in this series. One of the challenges with the slice and view tomography is that it can be difficult to verify that each of the slices are collected with uniform thickness. This will depend on the stability of your system. Things like temperature in the room can be important, and also how well the machine mills. One of the methods is to effectively cut lines and deposit them on the top surface, and then you can basically, if you put this like V-shape or cross X-shaped thing, you can measure the distance between features that you see within each slice. If you measure that distance, you can effectively get the distance and gray levels, and the geometry of the lines that you cut on the top, and so then you can work out how far through the thickness T that you've got. There's more descriptions in this beautiful book, Focused Ion Beam Nanometrology by Cox 2015, that shows you this idea. One of the issues we want to be able to also do is to look at site-specific tomography. We've just recently published a paper that gives us a large volume chunk lift out to enable 3D tomographic analysis. So this is site-specific FibSem tomography for EBSD analysis. This is really nice because it kind of links the tomography as well as the sample prep. Here we're effectively going to take our hexagonal piece of magnesium where we've aligned that the basal plane is in plane. We're then going to put an indent on the top of our object. We're going to lift out that indent in the residual deformation field. We're going to attach it to the edge of a grid. We're going to put the grid on the end of a post, and then we're going to do FibSem tomography to remove layer by layer of this slice. So we want to basically remove and tomographically reconstruct the 3-dimensional volume that surrounds this indent. Now the flow of the experiment is sort of shown down the bottom, and we're going to go through each of these steps to give us a flavour of how we did the tomography, which is basically enhanced with all of the sample preparation route that makes the analysis relatively trivial afterwards. And we're going to collect EBSD data is what's important. So one of the pieces that Ruth and I like to point out to people is these experiments are quite expensive in terms of instrument time. And so there's a huge amount of benefit from planning the experiments carefully. This indentation was done by Shuheng Li. And so we have a number of indents that were performed. We selected the correct indent to analyze. We did some EBSD data to verify the site-specific area. We calculated how much time it would take for the EBSD mapping. And we checked the sample could EBSD and produce beautiful diffraction patterns, etc. from our surface, so we could validate what was being done. Produced a series of to-do lists that could go through. One of the bits is these experiments take a significant amount of time, and you physically get quite tired as you do the experiments. And so therefore it's valuable to create simple to-do checklists to make sure that you have done each of the steps properly in turn. Here we have effectively the preparation of the lift-out coupon, and then we're going to do the FibSem tomography to do the slice-by-slice EPSD removal. And so here we're basically going to handshake the tomography wizard in the TestScan Essence software with the EBSD produced in the Oxford Instruments Aztec software. And then once we have collected all of this data, we do some post-processing in a combination of Python, Dragonfly and MATLAB to then enable us to produce a visualization of this structure. So the planning steps, this is sort of the overview as presented, but what we're trying to do in this process is to think, what do you want to know and why do you want to know it? One of the big questions here is if you can get away with doing anything without 3D, strong recommend. The 3D looks beautiful, but it's incredibly expensive and quite hard to do. So then you design the experiment. Is it feasible? How do you get the data efficiently? And the efficiently is both in time, but also in storage on the computer. We produce vast data sets for this. We do a series of trials to check milling performance for the tomographical slicing, but also in the sample prep. And so we start to work out how the fib currents and voltages will work in our material system across the right grains. And so actually we had a trial of a similar indent to verify this was the case. And we're also going to test the analytics on the fresh cross-section. So that enables us to verify that we can get good EBSD data once we've done the series of FibSem, because we really can't interrupt the tomography once it's running. And once we've finished the collection of the data, the next question is how do you make it relatively easy for you to understand? And so that's, can you collect the data that is segmentable, labelable, and can you store and analyze it relatively quickly? We're not going to spend too much time on that bit of the problem. It's a huge story in and of itself, but this is just giving you an idea of how the sample prep and tomography is going to be done in this experiment set. So we started off with some initial imaging. This is our indent. We have some deformation fields around the indent where the material has come out of plane. And so we did initial imaging and EBSD to show us the microstructure before we have done the fib work. And we've got the initial orientation of the HCP unit cell with regards to the indent and the microstructural regions. And here we have some deformation twins in our magnesium indent. Then effectively we have our array of indents, and we did our effectively deposition of tungsten or platinum, and here I think it's tungsten in this setup, so electron beam and then ion beam deposition to protect the surface of our region, so that then we can effectively enable the tomography to happen nicely without curtains, etc. as we go. So this is the deposition process, a nice little video to show you that. That's the I-beam deposition, I believe, given the height that's going on in here. So we're going to build up the deposition, and we basically can start to see the residual indent, the relief in that indent as presented. Once we've done the deposition, we're then going to effectively mill trenches, similar to our TEM-like lift-out. We're going to leave a fixed tab that we can then do a J-cut to release. We're going to do some undercutting of the region of interest, but we're going to create effectively some trenches to free it, and we're going to have a viewing trench to enable us to look underneath as we do the undercuts around trench 1 and trench 2. So we can then do the cuts. These are sped up slightly. We can do the cuts into the sample. And then we do polishing steps to enable us to get nice, deep, and good cross sections on the sides. So this is showing us multi-step milling, which is beautiful. Once we've done the trenching process, we can then do tidying up. So we're going to polish the side faces to enable us to remove the redeposition and clear up some of that artifact. So we can start to see that surface. This is a effectively lower current, is the more important detail, and we now have high aspect ratio to enable the redeposition to get out of the way. Once we've now done this, we can do the undercuts, and so the undercuts are performed to enable this cut to effectively free that the sample is now only attached by this edge. We basically do this is that we tilt the sample so that the fib is at an angle around 45 degrees that gives us the undercuts, and we're careful about the position of this cut with regards to the depth of the sample that we're removing. So the undercuts, you can't see them because we're looking under the SEM, but what we do see is the shadow of the cut as it's gone through the object. And so we see that cutting into this wall, we see that L-shaped black mark because the undercut has gone all the way through in the object. We can do the second undercut, and so I think, hopefully, there's the cut. Now here we were a bit too fast, and so we actually had ejection of a bit of material. It was out of the way of our experiments, but that wasn't ideal. So that's a little bit of debris that's not there. But we now effectively, that's come off because we've cut off this little bit of material. But we now have that this is a freestanding, except it's on this little edge, little tab we call it, and now we have that object. Then we're going to do the lift out process. We're going to bring in effectively the nano manipulator. So the manipulator comes in. We're then going to weld the manipulator. Just bear in mind quite how big our object is, but we've now welded the manipulator to the side of this. We're then going to cut the tab off. We're going to then clean up the edge, because this is the kind of the last bit. This edge is going to be what we are going to attach to our grid. And then we lift out the sample from the object. Once we've lifted out the subject, we can then move the sample out of the way, and we can look and insert the grid. So here's the grid. We've cut the end of the grid to give us a flat sample to weld against, and then we're going to position the sample carefully very close to the grid, and then we're going to do effectively the attachment process where we do an initial weld, and then we backfill in the object in here. Once it's nicely attached to the grid, we can then effectively cut the needle off, and then we just do a final extra deposition just to secure the object on the edge of the copper grid or moly grid. Once we've done this, we can then effectively do the tomography, and so this effectively is the object. We then are going to remove layer by layer, and then we can virtually reconstruct this object in Dragonfly using the EBSD data. So we have the band contrast and the EBSD data. And we've rotated all the EBSD data to give us a consistent frame of reference to enable us to directly compare with the top surface coordinate frame as presented. So this was the initial top surface EBSD. We've removed points that are poorly indexed, and this is the reconstructed top surface. And so we can see the reconstructed top surface, we get really good EBSD data, and we also have retention largely of the twins in that top surface layer. It's not quite perfectly reconstructed, basically because we need to choose that particular top layer surface, and there's been a bit of ion beam damage in the very top surface layers. But once we've done this, we can then effectively have the overall indent impression, the twins associated with the microstructure, and then we can, in our digital reconstruction, we can do cuts. So we've got the red plane that's presented here. And this example shows the presence of twinning and deformation, but it also shows that we didn't have an isolated grain, and so this was actually a fail from a mechanical perspective, but really nicely shows the benefit of the 3D data to reveal the fact that there is subsurface microstructure, and of course we can look at the grain structures, etc. in the 3D volume. So we have the red plane that's presented here, the ZX. We can see the green plane, which is a sort of half section XY plane. And then we effectively have the blue plane, which is a midsection here, a ZY plane, as presented. And we can go through a Dragonfly and pull out the planes to give us the EBSD data as if we collected that cross-section from our digital micrograph. And we can analyze this data in a lot more detail if we want. We didn't show this in this work because we're mainly focusing on the tomography approach and data collection in here. So we've now seen effectively Fib, SEM, and FibSEM EBSD data. We can also do EDS data, so energy dispersive X-ray spectroscopy, In this example, we're going to use a sample that was kindly supplied by a graduate student in London of mine, Yusen Wang, and this sample contains effectively tin bismuth. Tin bismuth is important for solders, and one of the examples for a lead-free solder system. In this, it's a low melting point material, which is important. It actually created some tricky difficulties in the FibSem prep, but you'll see it worked out in the end. So what's nice in the tin bismuth system, we effectively have two different Z contrasts, and so we can see in this secondary electron micrograph beautifully the microstructure that's presented here. When we set it up for FibSem, we can look in the bulk material and we can cut out a big enough trench such that the X-rays can be ejected and escape and be collected in our EDS detector that I think is sitting somewhere just behind. It's behind the fib. We also have a small shutter, so when the fib is running, the EDS is shuttered, the fib finishes, and then the EDS shutter opens, and then we perform the SEM EDS map. We have the tungsten cap on the surface, and effectively this is the the EDS, of that material, where we have the tin as the green region, and the bismuth as the red in this case. So we've got effectively this set up to do relatively fast fib and imaging, but the EDS took now a bit more time. It's 5 minutes to get us good quality data. We established this EDS by doing a series of trials to check how we set up the voltage for the SEM, the probe currents, the collection conditions for the EDS to enable us to get a good spatial resolution. This is about a 50 micron side width in the volume that we do. 115 side width in here. And we got about 436 slices in about 59 hours of FibSem time for this example data set. But it's beautiful. We've actually seen this micrograph elsewhere. And so here we can now start to see this microstructure, and we can see the value of the three-dimensional data set because you can see this herringbone-like morphology. And we can start to look at, for instance, the dendrite arm spacing, primary, secondary, and tertiary. We can also look at the decomposition of the eutectic, and we could look at the area fractions, and we could look at statistical regions. And of course, this data set is quite big. These micrographs are quite large for this tomographic data set, with extremely high contrast and very good segmentation for this data set. And so we can see and imagine the power of this approach. This was showing you that we can do it with EDS. Of course, the contrast from the backscatter is very nice as well, so bear that in mind. So hopefully at this point I've persuaded you that the tomography can be very valuable if set up. It can be quite expensive in duration, but basically we perform slice and view based approaches, and we are basically going to use fib-based cutting to produce a new free surface. And we're going to use imaging and analytics to collect the data and subsequent digital reconstruction. We need to be able to access the volume, so we typically do this, be careful about how we trench, put it on the edge of a sample, or do a full lift out. And what's not sort of to be underrepresented in this is that a huge amount of planning and data management is important for this. And really, I have to say, is that these are beautiful micrographs that we've produced, but each of them are very expensive. And so here, one of the questions is, is there an easier way to get to the answer that you want? And then you might, for instance, have one 3D tomograph, or maybe a couple that really sell the argument afterwards. But you can do quite a lot of useful information from two-dimensional cross-sections or appropriate surface-based approaches for this. So that's going to move us into our last lecture, where we're going to effectively then look at some future directions for FibSem approaches. And I'm also going to give just a brief outline of some capabilities at the University of British Columbia that may be useful for some of our audience.